this, all right? So one was E. coli. Do you remember, was E. coli facultative? Was it anaerobic, or was it a macroaerophil? E. coli was facultative. It can grow in both, all right? Then what about Micrococcus luteus? What was that? That was it anaerobe, or was it a macroaerophil? Macroaerophil, all right? Microaerophil, sorry. And then Clostridium is the one that's the strict anaerobe. So you're going to see Clostridium doing really well in here, right? And the other anaerobic or aerobic / microaerophil test was the phylloglycolate liquid, and that's what these represent. We don't want you to mix these, because if you agitate them, then you'll get everything all mixed up. So we should see the E. coli growing throughout. We should see the Clostridium growing only on the bottom, and the macroaerophil sort of growing near the top, but not quite at the top. Does everybody remember that?
Yes.
Okay. You might see these exhibits, and then you'll know. It's all going to be multiple choice, just like the midterm was. All right? So that's the format. I'm not sure how Gary and the lab crew are going to set up that latex for staff. Went through, you know, doing an inoculation with the streaking and waiting around. You would just add this substance to it, and then you would see an agglutination. And agglutination is just clumping. All right, so if we had this out, and we were to ask which one would not be an indicative of any agglutination, that would be what? Number 1. There's no agglutination there. You always use a control, all right, and then you would do the agglutination on your unknown, and if it matches the control, which is agglutinizing, then you know. It's really a no-brainer, all right? We also had the urea broth and lysine, which is to differentiate which two bacteria, Shigella, I'm sorry, not Shigella, Proteus and Salmonella, right? Because those two will do what on hectogen? They will produce hydrogen sulfide. So how can you tell the difference between them? Because they'll both get that, what, black color? You don't know which is which, so you got to go further with those. So you could use the urea broth, which is going to be positive for Proteus. It's going to make it pink. Or you could use the lysine, which is going to be positive for Salmonella, which will turn it sort of a purple color. So remember that those two tests are to differentiate Proteus from Salmonella and know which one is positive for the urea, which one is positive for the lysine, and it's right here, okay? You could also use the Entera tube we mentioned for this as well. This, I think everybody knows what this was all about when we put the various different, I think we used disinfectant, inoculated discs. We also used antibiotics. And we know that when you don't have any growth around the antibiotic, we call that the what? The zone of inhibition, which means that is it susceptible or is it resistant? It's
susceptible.
If we see that zone, it's susceptible. All right? If we see growth around the disc, then it's resistant. It's resisting the antibiotics' effects. I think everybody knows what this means, right? So I'm trying to go over it sort of in order what we did, and then we got involved with the EMB. And the EMB agar was selective for what type of bacteria, what category? The gram negatives, which were the E. coli, the Enterobacter, the Shigella, the Proteus, and the Salmonella. And then we also, so what did we see in the EMB? And I guess we could go over here now because we have examples, and I hope they're good examples. Because sometimes they're left out too long, and then they grow more stuff, and then we have a mess. Over there, sorry. And you can't miss that green metallic shine. You see green metallic shine, and we ask you what type of agar it is, you're going to know it's EMB. It's the only place you'll get that green metallic shine. So we can ask lots of questions. We could say, what type of agar is this? Green metallic shine, got to be EMB. We could say, what is this selective for? Gram-negative. And then we could ask, what is this differential for? E. coli and Enterobacter. Because E. coli is going to give you the green sheen. But if you get instead the pink one, which is not here, or this is it. No, that's not it. They got this confused over here. That's what I was afraid of. But anyway, the Enterobacter should show pink growth on the EMB. All right?
That's
So
that's
is that hectogen?
Yeah, the EMB is the one in the back probably, unless that's green.
Yeah, so this is for E. coli. And this one, this must be it then. This is EMB. This has got to be it, because this is EMB, and I'm not seeing green, so this has got to be pink. This is what you would see for Enterobacter. All right, does everyone remember that? It would be nice if we had had the right one here. I don't know why we have this here. This has got to be, what is this? This is Hect, and this has been here too long. Because it's like totally, I don't know what this is. I throw it as a frisbee. It's not a good example. But we know that EMB is differential for Enterobacter, pink growth, EMB, green metallic shine. Sear it in your brains. All right? And we get to the hectogen. These hold up better. With the Proteus and Salmonella, we get the black hydrogen sulfide appearance, because it's making that gas, ferments that. The Shigella is going to be the sort of greenish, aqua bluish, and I don't like this one. It's just not showing you what it should look like, and I'm wondering whether things got messed up or not. But on the test, we'll have ones that will be very clear.
And on the EMB, they should be all the same.
EMB?
Yeah, they should be all the same.
Yeah, well, if you have any of these three, Salmonella, Proteus, or Shigella, they're non-fermenters. You're not going to see any colors. You might see growth in colonies, but you won't see any colors because no fermentation will occur with Shigella, Salmonella, or Proteus on EMB. What will you see on the hectogen for Enterobacter or E. coli? You'll see that salmon color, right? That salmon color, which I think that's what they're trying to say this is, because that's hectogen and you got a salmon color. So if we see this, and we ask you what it could be, and one of the options, it's going to be the same, right?
Hector is either E. coli.
E. coli or Enterobacter is going to be salmon on hectogen. That's the possibility. All right, so that's the EMB and hectogen. And unfortunately, these particular examples, I like these two for Proteus and some Salmonella.
For EMB, that's supposed to be
EMB looks like this is EMB, and it's pink. This should be the Enterobacter. EMB, growth, no color.
Could be any of them.
So this could be Proteus, Salmonella, or Shigella. I'm imagining that they're trying to say this is Shigella because it's not black, but it should be more blue-green, and I'm just seeing, I don't know. I don't like this one. I wouldn't use that on the exam. If I'm going to use one on an exam, I'm going to use one that's definitely, no question, that green, which is closer to this. I don't like this one either. But we'll make it clear on the exam. It will definitely be blue-green as opposed to black. All right? All right, the Enterotube. Gary told me there might be a question using the Enterotube. Now, with the Enterotube, all of these colors, you got to look at them, because if we go from this to this, that means that this is a positive result. He says negative, positive. So, this is not the one. there's one that has numeric values on it. And it's not out. Well, if it's not out, then if it's on the test, I'm just going to have to throw that question out because I'm not going to make you responsible for something that they don't have out here. But just to let you know what it is, each one of these will have a numeric value. And if you see, for instance, this turn to that, that means it's a positive for glucose, all right? These are not right. These are not what we're going to see. So this isn't changed. All right, so this is not a positive, that's a negative. There's a numeric value for every one that changes. So that's changed, that's changed, that's changed, that's changed, that's changed, change, not changed, changed. You'd have numeric values, and then you would add up the numeric values, and then you'd have an index book to look up. And then the numeric value would be next to the bacteria. All right, because whatever is positive is going to be specific for that bacteria. So, I don't know, he didn't put them out. This is really an odd looking one. So, if it's on the exam and, ooh, this is it right here. Okay, now this is what we're talking about. All right, so you see, this is the enteric tube. Okay, this is positive, that's negative. See how that's positive? Yellow to yellow. That's not positive, stayed the same. That's positive. That's positive for the black. That's not positive, that's not positive, that's not positive, that's not positive. That is positive, that color right there. That's not positive, that is positive, that is positive. So every single positive one is going to have a numeric value. So you see the numeric values? This one is for that. That one is for that. All right, so these are both positive, so you're going to have 3 value here. Now, that's positive, that's not. So you're going to have only 4 and 2, so you put 6 there. And then over here, that's not positive, that's not positive, that's not positive. You have 0 there. Now over here, that's not positive, that is. You'd put 1 there. And then for these, not positive, 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 you put 2 and 1, 3 there. So you see, by taking these numbers, whatever this number is, whatever that number is, whatever that number is, whatever, you'd have a 5-digit number. Then you go to a book, you look for that number, the bacteria that is there, that is by that number is your unknown. See how that works? That might be on the exam. So this is a positive indication. If you see this color anywhere in here, you're going to give that number value. Okay? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. No. There are never going to be that many numbers that are going to show up. All right, I'll have to think about however they set it up, whether or not I'm going to let that be a fair question or not, because he should have put that out the day that we were going over it, instead of just putting it out now. All right, that's all there is to talk about here. Let's go back over here to the auger, the blood auger. And then the urine. Oh, also the catalase. Remember what we did with the catalase? The bubbles, what does that mean? It has the enzyme catalase, which is going to break down what? Hydrogen peroxide into harmless water and oxygen. And what particular bacteria had the ability to do that? Was it the staph or the strep? Staph aureus was the one that could do that. Has catalase to protect itself from hydrogen peroxide. It's good to look at this. All right, because this gives you a lot of information. You can find this on the biology website for microbiology. Does everybody know how to get there?
Yes.
All right, if I was you, I would be just going through that website, look at the pictures, find these flow charts. Everybody should be able to make sense out of this. If we do the catalase test, if it's positive, it's staphylococcus. If it's negative, it's strep. We go down. What do we see on mannitol salt agar if it's staph? we would see a yellow growth, right? Because it ferments. If it's Staph epidermidis, which could be, what are we going to see? Pink, all right? All right, so here it is, pink. Here it is, Staph aureus, yellow, on the mannitol salt. If you want to confirm Staph aureus, we can do the slide agglutination test. If it gets clumping, it's Staph aureus. That's the quick way, right? So that explains this leg. Streptococcus, we can go to hemolysis on blood agar. Strep pyogenes is beta hemolytic, so that means that we would see total hemolysis. I might have to use one of those.
Are you looking for that one?
Beta hemolysis. So that's beta hemolysis, right? So you'd have strep pyogenes or Staph aureus is beta, right? Okay. So how about strep pneumonia? What would you have with that?
Pneumonia alpha.
Strep pneumonia, you would have alpha, which means you have partial. But this one's not shown. Well, it is. It's a blood agar. Yeah. This one, it could be sort of like a greenish cast to it as well. Partial, you can't see all the way through it, and it's green. That's alpha hemolytic, and that would be the pneumonia, right?
That's the strep
pneumonia.
pyogenes.
That would be this one then. Why is it? Oh, here's, it's even more green there. See the green on the edges? It's not growing near the opticin disc because it's going to be sensitive to that. But you can see the green. It's not totally transparent. That's what it looks like right there. This sensitive to opticin. Remember that strep pneumonia is sensitive to opticin. The letter P means opticin. The letter is just one letter away from the antibiotic. So OP. P, one letter away from opticin. P for pneumonia. Kind of keeps it all together for you, right? The bacitracin is for the strep pyogenes, and A, one letter away from B for bacitracin. I remember that bacitracin is going to be effective on strep pyogenes because I think of strep pie, blueberry pie, B for blue, B for bacitracin. That's the way I always kept it straight in my head. All right, so no, the bacitracin is for strep pyogenes. No, the opticin is for strep pneumoniae. That's what this flow chart shows you, all that information, all in one chart. And this is the mannitol salt, which we went over. The blood agar. A lot of these are not great examples. This is good. The epidermidis is usually going to be gamma. But if you leave it there long enough, the toxins that are produced by the metabolism, because it's still going to live, it's just not going to hemolyze the blood, could cause some hemolysis. So that's why they say that the Staph epidermidis is anywhere from the alpha to gamma. All right, alpha might if it's there long enough because of the toxins produced, but if it's just been done, not a long period of time, it's going to show gamma, none, no hemolysis.
What about hemolytic growth for Staph aureus?
Hemolytic for Staph aureus, you're going to see beta.
Is it?
Yeah, beta.
Okay. Beta all the way through, right?
Yeah. So you're seeing that here. That's beta. You can see your finger right through it. So Staph aureus, strep pyogenes are beta. Strep pneumoniae is alpha, and the Staph epidermidis is usually gamma. But if it's left in long enough, it can actually do alpha. So those are the results of blood agar. Now here is the urine plates, and we use 3 different agar plates for urine. We use EMB, mannitol salt, and SAB. The chart that you see on the lab website shows you all the possibilities for the different bacteria results. What you got right there is perfect to study. So when you look at E. coli, it's a giveaway. You love E. coli because it's going to give you the green sheen. You can just stop what you're doing. It's E. coli. You see green sheen on anything, right? And you might see somewhat type of hemolysis, some alpha with E. coli. And you're going to see growth on the SAB. And remember, there's growth on SAB except for the strep pyogenes does not show any growth on SAB. That's another good fact to remember. The other ones will show growth on the SAB. Only strep pyogenes, no growth on SAB. The one on SAB that really sticks out is Candida. It's like clumps of ice cream. Different than what you see there, or this one you might get confused, but which one looks more like ice cream clumps? This guy here, right? So Candida albicans gives itself away on the SAB very, very clearly. The strep pyogenes gives itself away because there's no growth on the SAB. And on the blood agar, you would see beta hemolysis. So that supports strep. Why wouldn't it be Staph aureus? Because with Staph aureus, you see growth on the SAB. Okay, so you've got to, know all these different agars. Enterobacter, on the EMB, you should see the pink. I'm sure it's the EMB. Yeah, pink, Enterobacter, EMB. You do see growth on the SAB, but that doesn't tell you much. On the hectogen, this is the blood. Enterobacter aerogens, it doesn't look like it's slight. Maybe, I don't know.
Enterobacter for EMB?
Yeah, this is Enterobacter for the blood, and it's looking like it's alpha.
It says alpha.
Yeah, it looks alpha if anything, but it's not clear. So that's where, you got to be a little bit more like Dick Tracy. And I would stick with this one because this one, EMB, pink, and that can only be Enterobacter. So the more facts you know about all this stuff, the more ammunition you have to get it right. Because unfortunately, some of these plates, if they're left out too long, they spoil, and you see growth when you shouldn't. And then sometimes they're not done properly, and you don't see what you're supposed to see. And sometimes they could do a better job of putting some samples out here, if you ask me. But that's, we don't want to complain too much. What did we use this for? We used ultraviolet light. So you know that if you exposed it for 60 seconds, you're less likely to see growth, right? Because it's going to be killed. 30 seconds, if you see some growth, why would you see some growth? Because it didn't kill enough of it. There's still some alive and it can regenerate, it can repair itself. And the 0, you see growth because it wasn't exposed to ultraviolet. You remember what we did there. All right. All right, we covered pretty much everything except the ELISA wells. Do we have any ELISA wells out? Okay, so with the ELISA wells, if we're going to ask which sample, the top, middle, or lowest one, the person has had the virus in their system the longest, which one would it be? The top, middle, or lowest? Top. And if we were to say which person has had it the least longest, the bottom. And if we were to ask what is coating these wells? What is coating the well? It's the antigen for the HIV virus. Remember, the antigen is the identification marker that's on a pathogen. That's what your immune system doesn't recognize. If your immune system doesn't recognize it, it will make antibodies against it and clump on it. All right? So that's what they do with the ELISA is they have a special antibody that has an enzyme, and the enzyme they mix with also a substrate that changes color. So if the antibody bonds to an antigen, then the enzyme's there and it will change color. So that's why you get this. If the person doesn't have anything there, then they don't have the virus. If we see this positive, do we say stop, you have HIV, or do we do something else? What do we do?
Western blot
to confirm. That's pretty much all there is to know about this one. Okay. Remember what we did here? There were agar plates that were soaked in either nothing or something, right? And the one that showed growth was the coleslaw. So if we have this on, they might have these exhibits. To tell you the truth, I don't know everything that's going to be on this exam because the lab crew comes in here, and every semester they set up mostly the same kind of exhibits, but every once in a while they vary it. So if we did see this here, it would probably have a card that says, as per your lab, which one showed the pathogen. And it's to see, did you come to class? Did you pay attention? If you did, you know it's the coleslaw, right? And then they might have something, I don't think they will, but they might have something for the handshake. Remember what we did with the handshake? Yeah. We had an agar plate, and we drew a line down the middle, and then you took your bare fingerprint on one just to show that there's no growth there. And then if you're the one who's going to be the index case, you're going to take your other gloved finger and put it in the pathogen. And then you're going to put that on the right side of the agar plate, just to see if there's going to be growth there. Then you're going to take that finger, you're going to put it on the finger of the person that you're shaking the hand. Then you're going to take that and put it on the right side of their agar plate. Then on the other hand that is not contacting the person who's spreading it, they're going to take the glove off and do the left side to show the control. All right, so what we did there is exactly what I just described. So if you see one plate that's really got a lot of growth, and then another plate that's got some, which one is the index case? The one that's got some or the one that's got a lot?
A lot.
The one that has a lot. And then we would say, what type of process has occurred?
Transmission.
Right. All right. I can't think of anything else as far as what we covered from the midterm antimicrobials. A disinfectant, is that used on your skin or on an object? On an object. The antimicrobials would be used on your skin. Antibiotics would be used in your body. That was the first lab with the anaerobes. The next one was the enteric plates, the EMB, the HE plates. The next one we did the ultraviolet radiation. The next one was the mannitol salt, blood agar, and the bacitracin and opticin. The next one was catalase and urine streaking. The next lab was epidemic simulation. The last one was what we did last week, the ELISA. So we covered everything. All right? Okay, so now